We knew that the chronic renal disease caused the fibrosis of kidney, so the kidney lost its ability. While searching the therapy by two keywords, fibrosis and renal disease, we found the hepatocyte growth factor HGF can prevent the fibrosis of kidney. That research was really interesting to us, so we decided to study more about HGF therapy. Hepatocyte growth factor HGF consists of two chains, the alpha chain which is 69 kilodalton and the beta chain which is 34 kilodalton. The disulfide bond between of two chains produces the activation. When tissue is damaged, HGF is bind to c-MET receptor and occur many chemical reactions: cell survival, transformation, tuberogenesis, proliferation is occurred. In cultured myofibroblasts, major cell involved in fibrosis in many organs, HGF exerted inhibition of growth, suppression of fibrogenic cytokine expression, and enhancement of enzyme involved in subsequent apoptosis. In kidney, HGF stimulates DNA synthesis in cultured renal tubular cells, and it has a potent anti-apoptotic effect on glomerular podocytes. And after nephrectomy, HGF enhances growth of other kidney. We call it as compensatory renal growth. HGF is already widely used in the clinics. The research is deleted with acute kidney injury and dialysis. When doing dialysis, they used cellulose membrane CE and polymethyl acrylate membrane PMMA. After the dialysis, serum HGF level of group CE was higher than group PMMA after 15 minutes, 240 minutes, and recovery. It shows us that change of dialytic membrane might improve clinical outcome of acute kidney injury. But high concentration of HGF can cause renal cancer, gastric cancer, hepatic cancer, and stimulate the growth of cancer cells. This table shows that HGF prevents apoptosis by viral toxicity. ADV-CMV LAC-G is virus vector, does not produce HGF. ADV-CMV HGF is a virus vector, produces HGF. In this table, we can see the ratio of normal cell in the HGF vector is more higher than that in the LAC-G vector. And in the HGF vector, the ratio of apoptosis necrosis cell is more lower. Also, we know that as the virus concentration increases, apoptosis necrosis increases. So, table 1, we can see from apoptosis induced by a high concentration of virus HGF is shown a protective effect on renal tubular cells. Table 2 shows a protective effect of HGF on the apoptosis by hypoxia. Hypoxia is the most common form of acute renal failure. COCL2 is hypoxia-induced factor. In table 2, we can see the ratio of normal cell. Early apoptosis in the HGF vector is more higher than that in the LAC-G vector, and the ratio of late apoptosis necrosis in the HGF vector is more lower than that in the LAC-G vector. As a result, we can know that apoptosis induced by hypoxia is prevented or delayed by HGF expression. In these studies, HGF is thought to be able to act as apoptosis inhibitor. In the initial gene therapy, a viral carrier which is expected to transfect efficiently and express the gene has a limit in clinic because of acute systemic toxicity, humoral and cellular immune responses, mutagenic potential, the past number in gene size limitations. Therefore, now there is a study in the direction of developing non-viral gene carriers that targets a cell or to deliver a gene for long without systemic side effect, and the study is now progressing. The advantage of gene therapy: if we use general chemical substance, we can avoid the systemic side effect by simple diffusion. In addition, it only serves as relaxing the ill rather than treating. Gene therapy can activate or inhibit protein synthesis by introducing a specific cell or protein so that cell's function can be changed. OCMCH introducing a carboxyl group to a 6-carbon of the mixture to improve the applicability, and PLL with high efficiency gene are synthesis. LMWSC, lower molecular weight water soluble chitosan, is lowering cytotoxicity of the PLL, increases gene transfer efficiency of chitosan and synthesis of PEG and FA for the delivery system to accurately target specific cell producing. Pictures are listed in the order in easy to understand the synthesis of POCPF. The results confirm that the final product that has been successfully synthesized for POCPF by identifying all of the characteristic peak of the molecule synthesized. Depending on the content of PEG, FA increasing through the AP, it can be confirmed that the remaining amine does not form a POCPF well by reducing the complex. C is the result showing that POCPF degradation in the body and release the pDNA. D is the result showing that the POCPF is better protecting the pDNA from DNASD because of content of PEGFA and will form a complex if the 2%. Finally, POCPF was the result showed that have a useful value as a gene delivery system. Throughout the researches, we know about the gene carrier that permits effective usage of HGF in renal disease therapy. And we know that the research of the gene carrier synthesis is vibrantly proceeding to solve the problem by side effects. So what we have to do is finding new gene carriers which can make stable binding with HGF but don't have side effects.